to the screencast video or online lecture for biochemistry. This is the beginning of the unit on protein function. We're going to look in this unit at hemoglobin and myoglobin because those are the proteins that are the most well studied and therefore the best suited for examining as a case study for protein function. We talked in class about the terminology of a ligand being something that reversibly binds to usually a protein, but could be something other than a protein, usually a larger molecule. We usually think about a ligand as a small molecule and what it's binding to as a larger molecule. That's not always the case, but it is the case that we're going to investigate here. So you have a ligand, you have a binding site, and when the ligand binds to the binding site, that's going to be reversible. So it's going to be favorable or be made more favorable by all the kinds of interactions that we learned about in the non-covalent interactions part of the course. So things like hydrogen bonding, electrostatic interactions, and also the hydrophobic effect. Here's an equation plus a cartoon, but if you ignore the cartoon, then you just have an equation which says the P for protein plus L for the ligand, and that reversibly binds to give the protein ligand complex, PL. That's a rate constant in the forward direction of an association, little a for association, and in the reverse direction, a rate constant for dissociation, a little d for dissociation. You can write an equilibrium constant expression for the association, so that's a capital K, much like you would have done in general chemistry, the concentration of the products divided by the multiplication or the product, of the concentrations of both the reactants. And that's also equal to, as we talked about in class, the forward rate of the reaction divided by the reverse rate of the reaction. In biochemistry, we usually take the reciprocal of the association and call that the dissociation. And so the K for the reverse reaction, that's a dissociation constant. It is an equilibrium constant, but it's the equilibrium constant for the dissociation reaction. And we tend to talk about it that way, and that will become more apparent why we do that as you proceed through this section. There is a letter, either theta, as you see here, or sometimes capital Y, which is used to indicate the fraction of occupied binding sites. Here's an equation for that. It's the occupied sites divided by the total of the sites, theta or Y. I'm not going to go through the mathematics here, but if you look at some of those equations on this and the previous slide, you can also see a representation of the fraction of sites in terms of the concentration of the ligand and the dissociation constant. The concentration of the ligand divided by the concentration of the ligand plus the dissociation constant. And that constant is going to be given in units of concentration or molarity. You can see why that is. If you look at the equation for the equilibrium, you have, if I invert this, you have P times L over PL. Two of those are molarities which cancel out, leaving one more unit of molarity. So the dissociation constant is going to be in units of molarity, or typically micromolar or nanomolar, some really small concentrations of molarity in the biochemical range of concentrations. So when you make a graph of the fraction on the y-axis, that's the fraction of bound sites, versus the concentration of the ligand on the x-axis, that equation, this equation, takes the form of a hyperbola. The hyperbola starts at the 0 on this type of graph, and then it approaches an asymptote of 1. So it never reaches 1, but 1 is the maximum, obviously because 0 sites being bound is a fraction of 0, and 100% of the sites being filled with the ligand is a fraction of 1. And in this representation, when you look at where half of the sites are bound, exactly half, and you find out what the concentration of ligand is at that point, that is the dissociation constant. So the dissociation constant gives you an idea of how tightly the ligand is bound or how favorable that binding is. If that binding is more loose or less favorable, then your curve will be shallower and the concentration of ligand that is required will be much larger. Conversely, if the binding is tighter or very favorable, then this curve will be steeper. And that's not very good. Let's try that again. And then your dissociation constant will be much smaller. So the smaller the dissociation constant is, the more tightly that ligand is being bound to that protein or that active site. Another way of thinking of it is you need less of the ligand to half saturate the binding sites. In this lower case, I need only, let's say, 1 whatever unit that is. Maybe that unit is nanomolar. But on the weaker binding, I might need 5 nanomolar, or it could get super weak, and then you might need as much as 10 nanomolar, and it would go on and eventually get to closer to 1, but not in the range of this graph. So the dissociation constant is a measurement of how tightly the ligand binds, and the smaller the dissociation constant, the more tightly that is. When we are talking about myoglobin and hemoglobin, it's really hard to measure the molarity of the ligand because the ligand is oxygen, which is a dissolved gas, and so it's easier to measure the partial pressure of the dissolved gas, and that is indicated, since it's oxygen, the PO2. The metric system unit of that is kilopascals. In general chemistry, you might have used atmospheres or torr to measure that. Torr, which are millimeters of mercury. Torr is a millimeter of mercury. I know that's hard to get your head wrapped around, but it's the amount of pressure that would raise up a column of mercury to 1 millimeter. And 760 of those equals an atmosphere. So the equation works out the same, except for instead of concentration of ligand, you put the partial pressure of oxygen, and instead of a dissociation constant, you get something called a P50, which is the concentration of oxygen that gives you 50% of the saturation or 50% binding sites. That's enough to go through on the PowerPoint slides. So now let's investigate the mini activity based on this larger activity called oxygen binding proteins.
Your pre-activity asked you to look up some information about hemoglobin and myoglobin.
So make sure that you know some things about the similarities and differences between the structure of hemoglobin and myoglobin. For example, both are heme-containing proteins. Heme is an organic molecule that coordinates an iron atom in the center. On the different side, myoglobin has 1 polypeptide chain, but hemoglobin has 4, arranged in a tetrahedral arrangement. And in terms of their function, hemoglobin is primarily located in your erythrocytes or red blood cells. Myoglobin is typically located in your muscle cells. Hemoglobin transports oxygen from the lungs to, let's say, the muscle cells, but also other tissues. And myoglobin stores that oxygen in the muscle cells until it's needed for respiration. Here's some questions about PO2 and kilopascals. The fractional saturation or fraction of binding sites that are occupied we've already discussed. P50 we have already discussed. Estimate the P50 for the graph and draw how you would estimate it. So much like we did on the PowerPoint slides, we're going to find the fractional saturation at 0.5, and then we're going to read that directly off of the graph, like so. The graph has lines, so we can make sure we're not reading that at an angle. And this is a skill you'll need to practice, because when you are reading graphs, you want to make sure that you're reading it as precisely as possible. It looks to me like this point crosses exactly on that line, which is exactly on the 1 mark. And if you are then going to report the P50, you need to check the units, that's kilopascals, and you need to figure out how many significant digits to report. So you don't just want to say 1 kilopascal, but you probably want to say 1.0 kilopascals. Remembering that when you see a division marked on a graph or a ruler or any kind of laboratory instrumentation that's measuring something, you can always estimate 1 point past where you see the lines. I could for sure estimate to the nearest tenths place, so 1.0. It looks to me like there are lines on this graph at 0.25, 0.5, 0.75, and 1. So perhaps I could estimate even one more decimal place, but I'm satisfied with 1.0. If you just put 1, then what you are implying, if I just put 1 kilopascal, then I'm implying that I only know that to plus or minus 1. So it's somewhere between 0 and 1. I'm sorry, 0 and 2. That's a huge distance. But when I put the tenths place, then I'm implying that I know that number to within plus or minus a tenth. So that's between 9/10 and 1.1. So that would be more like in this range. I think that's more realistic. So that's why we have in general chemistry rules for significant digits or significant figures, because when you give a result to a certain number of digits, that final digit that's significant is telling you to what level of precision you actually know that value. So practice that. All right, let's move on. Question 5 says, when myoglobin is in an environment where the partial pressure of oxygen is the same as the P50, what does that mean about the myoglobin proteins? Draw a picture of myoglobin proteins with oxygen bound or not to illustrate your answer. So let me give a little explanation to that. You can just draw a cartoon. So you don't even have to be this specific. Like this is a picture of a myoglobin protein, but you can just do a cartoon, something like this, right? Myoglobin has a binding site and oxygen. So think about that. Pause the video now, take a minute, and in this space on your paper or on some blank paper, draw me a picture of, well, that answers number 5. So pause the video now and write your answer. All right, here's my idea. Here's your myoglobin protein. Here's a myoglobin protein. If you want to illustrate that half of the binding sites are filled, you've got to draw at least 2 myoglobins. Because if I draw 1, I either have to put oxygen in or I have to leave the oxygen out. So in order to show 50% bound, you have to show at least 2 of them, one with the oxygen bound, and I decided to show another oxygen not bound to the myoglobin. So this would be a picture that would illustrate a concentration of oxygen at which half of the binding sites are full. Does that make sense? Obviously, there are hundreds, thousands, millions of myoglobins, so the P50 is a statistical thing. Some of them are filled, some of them are not filled. At the P50, it's 50% filled. So let's move on to the next question. Now you have a better idea of what you're going to draw. When hemoglobin is in an environment where the PO2 is the same as the P50, what does that mean about the hemoglobin proteins? And draw a picture of hemoglobin proteins with oxygen bound or not to illustrate your answer. So now that you know what to expect here, you're going to have to make an extension. Again, commit to drawing something before you pay attention to what I have to say about it, because once you've drawn something, you have something to compare and contrast with what you drew with what I drew, and that's going to help it stick more in your head. So pause the video and draw something for yourself now. All right, here's one thing you could have drawn. Remember, hemoglobin is tetrameric and it has 4 binding sites. So in my fabulous picture here, I've drawn hemoglobin. You might remember that it has 2 alpha chains and 2 beta chains, 2 alpha subdomains and 2 beta subdomains. And I've shown only 1 hemoglobin molecule with half the binding sites filled. That's one way you could do it. If you drew only 1 hemoglobin, then you would have to draw it with only 2 of the binding sites filled. Now, if you drew more than 1 hemoglobin, there are different ways to do that. So if you only drew 1, I'm going to ask you to pause the video again and draw some more. And if you drew more than 1, pause and think about it for a minute, and then I'll give you another answer that's consistent with this. So give it a pause. Okay, so now I have 3 hemoglobins drawn. What do you notice? Did I do it right? Are half the binding sites filled? There are a total of 12 binding sites. 6 of them are filled, so that's correct. But if I look at hemoglobin, the first one, there are 2 filled, the second one has only 1, and the third one has 3. So it doesn't have to be all of them are 50-50. You just have to have a total of the binding sites filled. And so yes, this is consistent. So if you didn't have it like that, make sure you add that to your understanding, that the P50 for hemoglobin is about 50% of the binding sites, but that on any particular hemoglobin molecule, It might have 2 or 1 or 3, could even have 4 or 0, right? I could draw 2 more hemoglobins, one with 0 binding sites occupied and one with 4, and that would still meet this description. Why aren't your answers to these questions the same? I think we've discussed that pretty thoroughly. It has to do with the number of binding sites per molecule, right? So make sure you can write some good sentence about that. I'm not going to give you one, but I think you can come up with one. So let's go on with the next part. This one says oxygen binding curves for myoglobin and hemoglobin. And there's a key down here at the bottom. So hemoglobin is the darker curve and myoglobin is the lighter curve. The lighter curve and the darker curve. So based on that model, estimate the P50 for myoglobin and hemoglobin. In case you can't read that, let's blow it up just a little bit. All right. So that's zoomed in a little bit. And you have it on your paper, perhaps. If you don't have it on your paper, do it from the screen. But what I want you to do is to give me the P50 for each of them with the correct number of decimal places and the correct units. So pause and write your answers now. All right, how close are you to my values? So for myoglobin, I think that this line crosses the 0.5 line right smack in the middle of the 0 and the 0.5 kilopascals, and so I put 0.25 kilopascals. Now, could you have put 0.2 or 0.3 and not had an extra decimal place? I could maybe live with that, but I really feel like it's definitely between 0.2 and 0.3, so 0.25, I'm justified in that. Also, the line for the hemoglobin curve is not exactly on the 3.5 line. It's a little bit past, so I put 3.6. Now, am I justified in putting another place on that, another decimal place? So I put 3.60. Maybe. I feel like that's a little bit of a stretch, because then I'm saying I know between 3.59 and 3.61, but I stretched it on this one a little bit. So I would have been okay with this one if you put either 0.2 or 0.3, and on this one if you just put 3.6. Definitely not 3.5. If you're not looking at this closely enough to see, let me erase these lines right there. If you're not looking at that closely enough to see that that line does not cross exactly on the crossing point, then you need to work on your close scientific observational skills, because that's an important graph reading skill. In fact, let's zoom in a little bit more, even more. So look at this part right there. If you said 3.5, you're saying it crosses right on that point, and I'm saying it doesn't cross right on that point. That's a skill. Reading graph is a skill that you're working on. So I'm going with 3.6. Hope you agree. So let's look at this next question. The PO2 in the lungs is approximately 13 kilopascals. What are the fractional saturation values for each protein at this partial pressure of oxygen? All right, so practice your reading skills again. Look at the graph and read me off the fractional saturation values at 13 kilopascals. So let me zoom in a bit more, and we'll put it right there, and I'll let you read that. All right, pause now. All right, the 13 kilopascal line is this one, and so that's right in this region. Does it look like they're exactly the same? They're awfully close, but not maybe exactly the same. I'm going to say, like if I divide this in half, that's 0.95. So is that maybe 0.98? And that's 0.97. That's what I'm going to go with. So I'm going to say that the myoglobin is 0.98 and the hemoglobin is 0.97 at 13 kilopascals. Do you agree with that? If you said they were exactly the same, again, work on your graph reading skills, being very meticulous. Next question. It says the PO2 in the tissues, let's say the muscles, is approximately 2.75 kilopascals. Now, you're going to see different numbers for these. I think the PowerPoint slides say 4 kilopascals. This says 2.75. As you might imagine, it depends, right? If you have a muscle that's under stress and exercise, it might be lower because it's using up the oxygen, and if it's at rest, it might be higher. So for this activity, we're going to say 2.75. So now you're going to do the same thing. Instead of at 13, 2.75 kilopascals. And then we'll write those values down here. So pause the video and come up with your answers. Pause now. All right, so the 2.75 is exactly halfway between this 2.5 and 3 line. So that crosses right here, and that's about right there. See what I'm doing there? If I see exactly where that crosses the line there, and exactly where that crosses the line there, I think it's below 0.35. I think 0.35 is right there. So I'm going to say for the hemoglobin, I'm going to say it's 0.34. Do the same thing for the myoglobin, all the way up here, about halfway, and it crossed there, 0.92. Something like that. So the next question, based on the data in Model 2, which is that graph, and your answers to number 9 and 10, explain why hemoglobin is able to pick up oxygen in the lungs and release oxygen in the tissues, and why myoglobin would not be effective at this task. That says take 10 minutes. So give yourself some time, and during that time, you're looking at this graph, and you're looking at these answers that you gave. That you said in the lungs, you have these fractional saturations for myoglobin and hemoglobin, and in the tissues, you have these values. So I'm just going to leave it on this screen, but on your paper, write yourself an answer to why hemoglobin is able to pick up oxygen in the lungs and deliver it to the tissues, but myoglobin would be unable to do that solely based on this data. Pause the video and write yourself a good answer. Did you write your sentence? I haven't written mine yet. I'm going to give you one more chance to write yours, but I did sum up the results in this little table. Hemoglobin 0.97 fractional saturation in the lungs versus 0.34 in the tissues. Myoglobin 0.98 saturation in the lungs versus 0.92 in the tissues. If you haven't written a sentence, pause it and give it one more shot, and my sentence will appear in a few seconds. All right, so did you figure it out? Here's my sentence. Based on these fractional saturations of hemoglobin and myoglobin at the PO2 in the lungs versus in the tissues, hemoglobin can deliver 63% of the oxygen it binds. 0.97 minus 0.34 that's still bound in the tissues means it goes down to 0.34, and that's 0.63 or 63%. While myoglobin could only deliver 6%, from 0.98 saturation in the lungs to 0.92 in the tissues. So what do we learn from this? One thing that is a takeaway is binding oxygen more tightly does not necessarily make it better, right? Myoglobin is binding more tightly. If you look at its curve, it's much steeper, and so it binds much more tightly. Whereas the hemoglobin curve is much shallower, has a much larger P50, and binds more loosely. Binding more tightly is not necessarily good if your function is to deliver the oxygen. Because if you were going to use myoglobin for it, it would bind tightly in the lungs, but it still binds pretty tightly in the tissues, so it wouldn't be able to deliver very much of the oxygen in the tissues, whereas hemoglobin can. Number 12. If myoglobin isn't very good at oxygen transport, which it isn't, what is the function of myoglobin? And how do the oxygen binding properties of myoglobin allow it to effectively complete its function? So pause and think about that for a second. And after you have a good answer either written down or a good answer in your head, then unpause the video. I'm not going to write a sentence here physically, but let's just talk about it. What is the function of myoglobin? You want it to be able to store oxygen or accept the oxygen that's present or that comes from the hemoglobin so it can be stored there for later use. So it binds it more tightly than hemoglobin. If you look at the curve again, at the pressure that we're talking about in the tissues, the pressure in the lungs was 13.75, so that was in this area, and the pressure in the tissues was 2.75, so that's in this area. The hemoglobin binds a lot and can then release it. Well, if you release it in the muscles, but you have myoglobin present there, it's able to bind to that oxygen and take it up. And then, of course, it depends on the relative amounts of those. So if you have a lot more myoglobin present, then it can bind a lot more of the oxygen as well. The fact that it binds more tightly than hemoglobin is going to be a good thing for its function, because it will be able to take the oxygen that's released from the hemoglobin and take it up. So perhaps you have some good sentence written for that. I hope this has been helpful. That's the end. Cheers.